Sorry, just had to sneeze there. Welcome to photosynthesis. In this plant, photosynthesis is occurring. You've heard of it, the process which allows photoautotrophs to turn light energy into chemical energy. Let's get into the real details of photosynthesis. For each plant cell, there are organelles called chloroplasts. In chloroplasts, photosynthesis occurs. Well, that's pretty much all you need to know for photosynthesis. Thanks for watching. It's a good thing Mrs. Magrino didn't see that. I'm sorry. There's a lot more to photosynthesis than just chloroplasts. But it sure is a good place to start. In the chloroplast, there are little pancake-like structures called thylakoids. The thylakoids are where the light-dependent reactions occur. There is also the stroma, where the light-independent reactions occur. The light reactions are when light energy is transformed into the chemical energy of ATP and NADPH. First, a pigment molecule in the light harvesting complex absorbs a photon of light. This energy is passed from pigment molecule to pigment molecule until it reaches chlorophyll P680. P680 then excites the electron to a higher energy state. The electron is then captured by the primary electron acceptor. Water is also split in photolysis. The electrons are supplied to chlorophyll P680 to replace the electrons sent to the primary electron acceptor. The primary electron acceptor kicks off what is known as the electron transport chain, which provides the energy for synthesis of ATP by chemiosmosis. Chemiosmosis is only possible due to the cytochrome complex, which pumps hydrogen ions against the concentration gradient into the thylakoid space. Sorry, there's an earthquake at my house, but only my house. The electron transport chain makes its way to photosystem 1. It excites an electron of chlorophyll P700, and an adjacent primary electron acceptor captures the electron, and an electron that reaches the bottom of the electron transport chain from photosystem 2 replaces the lost electron in P700. To do this, light energy is passed from chlorophyll molecule to chlorophyll molecule. Once an electron is passed up to the primary electron acceptor, a small electron transport chain occurs, which then powers the reduction of NADP plus to NADPH. Do you remember the cytochrome complex? How it pumped the hydrogens across to the thylakoid space? Well, ATP synthase will use these hydrogens to power oxidative phosphorylation.
The Calvin cycle utilizes the products of the light-dependent reactions.
It occurs in the stroma. 3CO2 combined with 3-ribulose bisphosphate utilizing Rubisco. Ribulose bisphosphate and the carbon dioxides make phosphoglyceric acid. Of them, 6 ATP from the light-dependent reactions come and are consumed by the Calvin cycle. Then, 6 molecules of NADPH are oxidized. This results in 6 glyceraldehyde 3-phosphates made. Only one of the 6 G3P will be used to make glucose or other compounds. The other 5 are recycled back using 3 ATP to make ribulose bisphosphate, which will be used again to combine with the carbon dioxides. Okay, the plant has made its glucose, but let's say an animal eats that plant. How does the animal utilize the energy of glucose? Well, in order to do that, cellular respiration is utilized. The first step of cellular respiration is glycolysis. An ATP molecule energizes glucose, turning it into glucose 6-phosphate. Once glucose 6-phosphate is formed, it then turns into fructose 6-phosphate. Another ATP comes and gives its energy to turn fructose 6-phosphate into fructose 1,6-biphosphate. The fructose biphosphate then splits into 2 glyceraldehyde 3-phosphate molecules. A phosphate group attaches to the G3Ps, and NAD plus is reduced into NADH. This creates biphosphoglycerate. Next, adenosine diphosphate comes and turns into ATP. This yields 3-phosphoglycerate. The phosphate group then moves to the 2 position to make the phosphoglycerate 2-phosphoglycerate. Water leaves the phosphoglycerate molecule. This yields phosphonol pyruvate. ADP comes and turns into ATP. This yields the final product of 2 pyruvate molecules. The pyruvate molecules from glycolysis are what are oxidized during this process. NAD plus is reduced into NADH, and a carboxyl group is removed from pyruvate and given off as a molecule of CO2. Meanwhile, a coenzyme A combines with this pyruvate molecule. This creates a 2-carbon molecule joined with a coenzyme A. This is abbreviated to acetyl-CoA. 2 NADHs are made due to the pre-Krebs cycle. The citric acid cycle starts with an acetyl-CoA molecule. The coenzyme A breaks off and 2 carbons enter the cycle. These 2 carbons combine with a 4-carbon molecule called oxaloacetate to create a 6-carbon molecule called citrate. The citrate then reduces NAD plus into NADH. CO2 leaves the cycle, and a 5-carbon molecule called alpha ketoglutarate is made. Then CO2 leaves the cycle again, NAD plus is reduced to NADH again. ADP is phosphorylated to ADP. This creates the 4-carbon molecule succinate. Then FAD is reduced into FADH2. This creates the 4-carbon molecule malate. Then NAD plus is then reduced into NADH. Each citric acid cycle creates 3 NADH, 1 ATP, and 1 FADH2. This is done twice, however, so in total 6 NADH, 2 ATP, and 2 FADH2 are created per glucose. Before this point, little ATP has actually been made. However, a lot of electron carriers have been. These are going to be used in oxidative phosphorylation. This step occurs in the cristae of the mitochondria. NADH from the electron carriers is oxidized into NAD+. This creates an electron transport chain which powers the active transport of hydrogen ions against their concentration gradient. Throughout the cristae, there are electron carriers. After NADH is oxidized, FADH2 is as well. The electron transport chain goes from electron carrier to electron carrier. More hydrogen ions are pumped into the inner membrane space. More electron carriers are evident throughout the inner mitochondrial membrane, or cristae. Eventually, the electrons from the electron transport chain combine with 2 hydrogens and 1 oxygen to create water. However, ATP still has to be made. To do this, ATP synthase is in the cristae. Remember the hydrogen ions? These go through ATP synthase to power oxidative phosphorylation. In total, oxidative phosphorylation can power up to 38 ATP molecules. And that's how lettuce or plants or anything that uses photosynthesis becomes available to humans, animals, and all of us. Thank you for watching.